Hello, and let's begin our discussion of cells, part one. But first, I must apologize for the sound quality. I did receive my Wazo Snazzo new microphone. However, there was an adapter that was needed, so that has now been ordered, and I should have that this week probably. So let's just move right into cells, and we'll just muddle through the terrible sound quality. All right. There are levels of organizations that we have, the cell being the smallest unit of life. Tissue is where we group a group of these cells together, the same cells, and these make up a tissue. An organ is a group of tissues that make up an organ. So there's a group of similar tissues, which of course are made of similar cells, and these make up an organ. And then finally, you have your organ system, where you have several organs that make up a system. Let me give you an example of that. Here we have a muscle cell. You can see over here, there's a muscle cell, one muscle cell. We put them together, group them together in a tissue, and it makes, for example, the smooth muscle tissue. The organ, you put that smooth muscle tissue together, combined in one area, and you'll have your stomach, which is an organ, as you can see, and then you put the stomach and all the other organs that make up the system together, and you have the digestive system. So again, the levels of organization are cell, tissue, organ, organ system. Words that you're going to want to know when we're talking about cells. First of all, unicellular. That prefix uni means one. So if something is unicellular, it means it only has one cell. And examples of that are certain types of bacteria. Multi means many, and so a multicellular, we're referring to that it is consisting of many types of cells. An example of a tree, a human, a dog, anything that has more than one cell is multicellular. And then you have subcellular. Sub means below, and cellular, subcellular, is it lacks the organization of a cell. All right, so different classifications of cells. The first one being eukaryotic. A eukaryotic cell is a cell that has a true nucleus. This means that that nucleus is wrapped in a membrane. And so it's got the genetic material, and that's wrapped up in a membrane. Plant or animal cells are eukaryotic cells. They are more complex. Not only do they have a true nucleus, but they have membrane-bound organelles. So when you hear Golgi body or lysosome or ribosome, anything like that, those we're talking about a eukaryotic cell because they have membrane-bound organelles. A prokaryotic cell does not have a true nucleus. All bacteria are prokaryotes, and so they don't have a true nucleus, and they do not have membrane-bound organelles. They're very simplistic organisms. They're less complex. Now, DNA is, the DNA of bacteria is circular. You can look here, look at the difference between the prokaryotic cell. Here you have your prokaryotic cell, and it has a cell membrane, and it has genetic material inside the cell. And then the eukaryotic cell here has a true nucleus. It's got a membrane surrounding it. There would be DNA in here, but you have these different Golgi body and mitochondria and all these different membrane-bound organelles within the eukaryotic cell. And of course, there we have the different organelles. Okay, we need to know a little bit about the history of the cell. Whoops, let's back it up just a little bit. Whoa, what's happening here? Let's go back. Okay, back to here. The history of the cell. We have Robert Hooke. Now, Robert Hooke used a compound microscope. He is credited with using the compound microscope. He looked at a thin slice of cork of plant material, and he saw these little things in there, these little square things, and he called them cells. So you can see, this is what he saw, these rectangular things in here, and they looked like cells to him. Which we know, of course, plant cells do have a rectangular shape. Another part of the history, another big player here is Anton van Leeuwenhoek. I love to say that. Anton van Leeuwenhoek. And what he did is he observed cells in pond water. And what he found is that cells are everywhere because he saw them everywhere. And Leeuwenhoek is credited with developing the first microscope. And Schleiden and Schwann, what they found is that all plants and animals are made up of cells. Finally, we have Virchow, that all cells come from existing cells. They are coming from somewhere, and he found that they were coming from already existing cells. And there are some protists in there. Okay. Now, all of those individuals are important, and their discoveries are important because they led to what we now currently use as the cell theory. The first part of that is that all living things are composed of cells. Everything that's living is made up of cells. And secondly, cells are the basic unit of structure and function in living things. And then finally, in the third part of the cell theory is that new cells are produced from existing cells. So they come from pre-existing cells. All right, what we also know is that all cells have a cell membrane and they have DNA. Whether they're eukaryotic cells or prokaryotic cells, they all have a cell membrane, which is this outer covering that separates the inside from the outside. And we're going to look at that cell membrane in a bit more detail this week. And then you have your DNA. They all have the DNA, the genetic material. Okay, moving on into the cell organelle and cell parts. Okay, the nucleus. This is the brain of the cell. It controls all of the cellular activity. And you have to have this membrane surrounding the nucleus. So you have a membrane, just like you have a cell membrane, you have a nuclear membrane, a membrane that encloses that nucleus. And it controls what comes in and goes out of that nucleus. And you're going to see, especially once we get into protein synthesis, you're going to see why that nuclear membrane is real important there. And then, of course, the nucleoplasm, very similar to the cytoplasm within the cell, and it's the supporting substance that's inside the nucleus. All right. Now, this chromatin, this is the genetic material that's inside the nucleus. Looks kind of like spaghetti in there. It's made up of DNA and protein, and it's this chromatin during cell division that becomes the chromosome. So when we study the phases of mitosis, when we look at that, we're going to actually see this chromatin move from chromatin to chromosomes. And then inside that nucleus is what's called a nucleolus, or a little nucleus. And it's here, very important, it's here where RNA is produced and where the assembly of those ribosomes begin, right here in the nucleolus. And you have your centrioles. These centrioles, that's a picture of them right down here. And they will produce the spindle fibers during cell division. Again, when we go through the phases of mitosis, you're going to see in one of those phases where the spindle fibers are produced. It's in these structures right here where, and you can see, even just looking at them, you can see that these tubes here, that you're going to have things that are produced out of there. They're set in a very specific format. Now, these centrioles are not found in plant cells, and as we move toward that, I'll explain to you more in depth. I am going to hold off on the rest of this, and we will cover the cell membrane in a little more detail coming up. That's all we have for today. Have a marvelous